This lecture is about translation, and translation is defined as protein synthesis. So you should have already learned about how the DNA is transcribed into RNA, and in eukaryotes, that RNA is processed so that a cap and a tail is put onto the primary transcript, and then the introns are removed or spliced out of the mature messenger RNA. That messenger RNA leaves the nucleus and goes to the cytoplasm and binds to a ribosome. And this is where we're going to talk about how the ribosomal pieces come together and the tRNAs carrying the amino acids come to make a new protein or polypeptide. So again, there are some good translation animations that you could take a look at. But we're going to first focus on the central dogma of molecular biology. So you know that DNA encodes for messenger RNA, which then encodes for the protein sequence. And what you need to remember is that the genetic code is basically three bases, three nucleotides that encode for one amino acid. And the genetic code table is shown here. Please do not try to memorize this. This will be given to you on the test. But do remember the genetic code table translates what one messenger RNA codon encodes for in the amino acid, like which amino acid is encoded. Okay, so it's the mRNA, and that mRNA is read 5' to 3', and the amino acid is then laid down. Okay, so here's a practice problem that will be similar to something on the test. I'll give you a DNA. You have to figure out what the other DNA strand is using complementary base pairing. And then once you have a DNA strand, you would then also try to figure out what the mRNA transcribed from that would be. And then you would have to read the mRNA from 5' to 3'. You want to look for the very first start codon and then be able to translate the amino acid sequence after that. Okay, the protein is always laid down amino terminus to the carboxyl terminus. Okay, so one way to help you remember is if you're doing translation, you are converting one language into a different language. So you're converting the RNA, the language of nucleic acids, to the language of proteins. And these are two different languages. They're made up of different alphabets. They're read differently. So think about if you were reading English, you would read English from left to right. Whereas if you were reading Chinese, you would be reading the words right to left from top to bottom. So it is a completely different way to look at the different molecules. Okay. So for the discovery of the genetic code, one of the things that they had to think about is how many nucleotides actually encode for each amino acid. If only one of the nucleotide correlated to a codon, that means there's only 4 nucleotides, so there's only 4 amino acids possible. And that is not possible since there's 20 amino acids. So what if one codon actually was made up of 2 nucleotides? Then for each of those 2 positions, you have 4 nucleotides possible. So it'd be 4 times 4 gives you 16 different amino acids. Again, that is incorrect because we have 20 amino acids. So then they tried a triplet codon, and that means there's 4 possible nucleotides at every position in the codon. So you would get 4 times 4 times 4, which gives a total of 64 amino acid possibilities, and that works. Since it's over 20 amino acids, that's correct. possibility. And then they did some actual research projects to show that it had to be in groups of 3. Okay. So one of the major techniques that was determined after they figured out it was a triplet codon is they used the cytoplasms from a bacterial extract, and they saw that and they put in radioactive amino acids into the same test tube, and then they threw in synthetic messenger RNAs and then waited to see what types of polypeptides were going to be made from that synthetic messenger RNA. They didn't know what was in the translation system, but they knew it worked. And this actually led to the whole genetic code discovery. So I have a little practice problem here for you to take a look at. So if you try this and it really doesn't make sense to you, don't worry about it. But if this, just think of it, when they did the in vitro translation system, it was pretty much a big puzzle. And so they would try different synthetic messenger RNAs and then look to see what kinds of polypeptides would be created. One of my professors was part of this project, and he said they did this at Woods Hole Marine Biology Labs, which was a super think tank. And if you're in Maine, what kind of creature would you work on? Lobsters. And so they would go and buy fresh lobsters in the morning, chop off the tails, save that for dinner, and then they go and work on the heads to get all the tissues for their experiments. And then at night, then they could have a free lobster dinner paid by the government. So anyway, so hopefully, if you take a look at my practice problem, this kind of makes sense. It shows you the logic, the step-by-step logic of how they came about to figure out the genetic code table. Okay. So this is a different expression of the genetic code table, and it shows you for every one of the amino acids how many codons are possible. So you can see that some amino acids, there's only one possible codon. Like look at tryptophan and methionine. For others, like arginine, leucine, serine, there's actually 6 different possible codons. So anyway, so this particular genetic code table can be used to show the abbreviations, show the amino acids that are abbreviated in 3 letters, or they could be abbreviated in 1 letter. Okay, so for my class, we typically do the 3-letter ones. Okay, so you know that there's 3 different kinds of RNAs, the main types of RNAs, and one of them is called the transfer RNA, the little tRNA. And their main job is to carry the amino acid to the ribosome. So there's 2 important places on this transfer tRNA molecule. The 3' end is where the amino acid will be attached. And then at the bottom of that tRNA molecule, you'll see what looks like the anticodon loop. And what you need to figure out is this tRNA has to bond to the messenger RNA codon. Okay, so the messenger RNA and the tRNA anticodon are complementary base paired, and they're also in anti-parallel orientation. So if you're going to have a messenger RNA that is reading AUG, then what is the tRNA anticodon of that? And then you would go look at the genetic code table to figure out exactly which amino acid that's carrying. Okay, so why don't you try this for this particular tRNA and figure out how do you read the anticodon, which messenger RNA codon would it match to, and then which amino acid should it carry. And then here's the answer for you. So make sure you try it out. The third type of RNA, so again, we had three types. So we have messenger RNA, the transfer RNA, and then third type is ribosomal RNA. And the ribosomal RNA is actually just part of the structure of the ribosomes. So ribosomes come in two parts. There's a large subunit and a small subunit, and then they come together to be the complete ribosome. So both prokaryotic and eukaryotic ribosomes are different in the terms of size and how many proteins and how many RNAs are involved, but you just need to know that there's a large and a small subunit for this class. Okay, when you put the ribosomes together, it actually shows that there's 4 binding sites. So one binding site is for the messenger RNA. One is called the A site, P site, and the E site. So the easy way to remember is the A site is going to bind to a tRNA that carries 1 amino acid. So think A for incoming amino acid. Then the P site is to bind the tRNA that is carrying the peptide. And the E site is to bind the tRNA that is empty and ready to exit. Okay, so that's the ribosome. And again, there's many different animations to help you follow the process of translation. And I do think that watching the process continuously does help. So here's the overview of the translation process. We have all the pieces that have to come together at the initiation step. And then there's the elongation step where we keep adding more and more amino acids together. And then we have a termination step where everything will disassemble and the protein will be floating away in the cytoplasm. So this picture is showing you the initiation of translation in a bacterial cell. And you can see that at the top, there's a small ribosomal subunit that will bind to a messenger RNA. And then there are transfer RNA. The very first transfer RNA will come and bind. And the close-up shows you that the first start codon on the messenger RNA is always AUG. So that's 5' AUG 3'. So therefore, the tRNA that's a complement of that has the anticodon 3' UAC 5'. So that first tRNA is going to bind, and then the large ribosomal subunit actually binds. And when it's complete, you can see that the first tRNA is actually in the P site, not the A site. So this is an exception to the rule that the A site is for incoming amino acids. Okay, basically it has to be here in the P site because otherwise you'll never have an empty place for a new incoming amino acid. Okay, so now this is the close-up for the second amino acid coming in. And you can see now the amino acid 1 is in green, that green circle, and the amino acid 2 is the blue circle. And what's going to happen next is you'll have the first amino acid get transferred on top of the second amino acid, and then you'll be left with an empty tRNA and then a tRNA carrying the peptide. And of course, the 2 amino acids are joined together by the peptide bond. Okay, and so what's going to happen is everything will then have to move over. Okay, so this slide shows you all the steps in more detail. So just want to remind you, there's the enzyme called peptidyltransferase, and that's the one that actually joins one amino acid to the next amino acid and forms that peptide bond. And then translocation occurs to move the whole mRNA and the ribosome, one codon down. Okay, you repeat this process over and over and over. So every time there's an empty tRNA, it moves over to the E site. The tRNA carrying the peptide will move to the P site, and then there will be an empty spot for the brand new tRNA carrying one amino acid coming in. You keep doing this until you have a stop codon, and once you have a stop codon, there are no tRNAs that correspond to that, and so when the peptidyl transferase moves the peptide onto the next place, hoping to form the new peptide bond, there's nothing to bind to it. And so since the protein's already been released from the tRNA, they basically everything will disassemble away from each other. Once they disassemble, as long as the mRNA finds another ribosomal subunit, then it will start the process again. And so sometimes you'll use this one messenger RNA for 6 hours or 24 hours or 48 hours until it gets degraded. In certain tissues, you sometimes will need protein synthesis to occur very, very quickly. And so what you can see is that here is one messenger RNA, and you can see that there's actually quite a few ribosomes that have all been attached to this messenger RNA and starting to do the translation. And so you can see how at the very beginning, near the start codon, the peptides are very short, but as they move on to the end or the stop codon, the peptide is complete. Okay. So we've just gone through translation or protein synthesis, and you can see here these are all the different steps that can be regulated for protein synthesis or gene expression in eukaryotic cells. So if you think about it, every cell in your body contains all the genes that it will ever need. So if you were a liver cell, would you want to make the eye gene or eye proteins and the gut proteins and the blood proteins, or do you only want to make the liver proteins? And if you want to make liver proteins, at what point do you stop the gene expression of all the other genes? Do you make the primary transcript and then stop? Or do you make the primary transcript and then do the RNA processing and then stop and don't allow the RNA to go out of the nucleus? Or do you go through the entire making this mature messenger RNA, letting it go out to the ribosomes, and then go out to the cytoplasm and then degrade it? Or do you allow the protein synthesis to occur and then you degrade the protein? At what point would you stop this process if you were a liver cell and you didn't need all the other proteins encoded by the other tissue? All right, so think about that. And then our last topic is mutations. So we're going to be talking about point mutations. These are mutations that occur. They could be sporadic. They could be just, a simple mistake during the DNA replication process, or it could be something that was caused by a carcinogen or ultraviolet light. Okay, so what we're trying to show you here in the top panel is that there's a protein that's going to be made and happens to have 4 amino acids: methionine, proline, threonine, arginine, and then there's a stop. So if you happen to have a mutation and it causes that second codon to go from CCU to CCC, what's the effect on the amino acid? and hopefully you see they're exactly the same. And so this is called a silent mutation. So even though there was a DNA mutation, it has absolutely no effect on the protein's function and shape. But on this next protein mutation, it's called a missense because what happened was you changed the fourth codon from CGC to CAC, and that changed the amino acid from an arginine to a histidine, and that's a change. And so a missense mutation refers to a substitution in one amino acid of that protein. On the bottom panel here, you can see that the UAU codon was changed to a UAA codon, and that UAA codon happens to be a stop codon. So the result of this is that you end up with only 2 amino acids instead of 4. So you only end up with half of the protein that you were supposed to get, and that is bad. That protein will not have the correct shape and not be able to do the correct function. This is called a nonsense mutation when there is a stop codon that occurs too early and truncates or shortens that protein's length. Sorry, there's one more. Okay, so this slide tells you the hemoglobin mutation. So hemoglobin mutation, you can see that at the top side it says glutamine, GLU, and then on the bottom it becomes a valine, VAL. And that's a big change because glutamate is actually a negatively charged, it's an acidic amino acid, and valine is a hydrophobic nonpolar amino acid. And because they're completely different chemical natures, that causes the proteins to interact in an abnormal way. So you can see that it causes long, sticky protein tetramers, which then which then causes the blood cells to form a sickle shape. Okay. All right, so I made this up to help you with the idea, the big concept. Okay, so again, it doesn't. Anyway, so suppose our protein sequence was supposed to be the big bad dog bit the red cat. But if you had a missense, you can see how the cat changes into a rat. And if you have a nonsense, you can see how the sentence was cut and was shortened too early. And the last mutation we wanted to go through is called a frame shift. And so what happens with a frame shift is if you add or delete a particular nucleotide, it will shift how the entire reading frame is read. And then that entire sentence or entire protein sequence will be completely wrong. And so that shows how it could be a very bad thing. All right. On the left, again, I just picked up some pictures of normal red blood cells versus the sickle red blood cells. And on the right side, I'm trying to show you the mutations known for a particular gene called tryptophan synthase. And so let's take a look. And amino acid number 15, there's a lysine that is now changed to a stop. Okay, so in the original protein, it should have about 250 amino acids. But if you had a stop at number 15, that means you would only make a 15 amino acid protein, and that, of course, is way too short. It cannot have the same shape as the original protein, and that would be a big problem. So what is the name of this type of mutation? And then go through and see if you can figure out the names of all the other types of mutations listed here. Okay? The last picture here is to show you about post-translational processing. So sometimes proteins are made through translation, and then they're sent to another place before they actually start their work. And sometimes you don't actually want the protein to start working right away. You want it to be sent to a different place, to its final destination, and then start working. So, for example, our pancreatic cells make the digestive enzymes that go into the small intestine to work. And you would not want those proteases to start working in the pancreatic cell or in any of the ducts as they travel to the small intestine. So what happens quite often is the proteins are actually made too big, and they might have an extra piece. So let's look at the section on A or B. You can see that the proteins that was first made is too big, and then when it's sent to the final destination, there's a piece that's removed, and then you have the final proteins. Panel C here shows you that sometimes you make the proteins in pieces, and then you send both of the pieces to the final destination, and then they get cut and joined to become the final protein. All right, so that's the end of our translation